Okay, hi, this is Andy DaSilva, and for my oral presentation, uh, it, it is entitled Mechanisms of Antibiotic Removal, a Review. Why am I doing this literature review? For two reasons, for personal and professional. Personal because I have a general interest in human health because I like to understand how I can take care of myself uh, and I like to make sure that I stay fit and stay well. I don't like to get sick, obviously. And then I also have a daughter that missed school quite a lot last year due to different illnesses. She missed about 30 days of school. So it's really important for me to understand what it takes to be healthy and to understand also the implications of drugs such as antibiotics. And then I'm also an engineer at a wastewater treatment plant, so I thought professionally this would be a good topic for me. Abstract. So antibiotics are used to destroy or inactivate harmful bacteria. Antibiotics are very common for treating human health as well as animal health. But because they're so common, so popular, they are incredibly overused and overprescribed. And whatever's not used up ends up in sewage waters and goes into treatment plants. Currently, residuals from antibiotics are not regulated, so they can be found in biosolids and with treated effluent. And the reuse of sludge in agriculture applications and exposure to the aquatic environment is considered a major health issue and as well as an environmental issue. In addition, bacteria are constantly developing ways to resist antibiotics. So for my paper, I conducted a literature review on how antibiotics and drug-resistant bacteria are removed through the wastewater treatment plant processes. This is how my paper is organized. I started with the abstract, which I touched upon. Then I follow up with an introduction, and in part one, I talk about antibiotics. I give a background on antibiotics, and I go into physical, biological, and chemical mechanisms of removal, and then I finish with a conclusion. Introduction. Some highlights from my introduction. So I mentioned how antibiotics are used also, and I think everybody has probably used antibiotics at least once in their life, but they're also commonly used for livestock production. The reason for this is to promote animal weight growth, so they're combined with feed additives. And over half of all antibiotics actually goes towards animal production. And not all antibiotics are metabolized by human and animals, so sewage waters are the pathways for antibiotics that are contained in human feces and animal manure. And wastewater treatment plants, they're a hotspot for the accumulation of antibiotic quantities. So that's what it means is that antibiotics are accumulating in wastewater treatment plants. They're becoming hotspots. And because treatment plants rely on biological processes, there is a concern that they also could be promoting the growth of antibiotic resistant bacteria. So antibiotic residuals, they're hard to detect. Sometimes they're undetectable because ranges can be as low as a few nanograms per liter. So just because they're not detected doesn't mean it's not in the source material. The source. It doesn't mean it's not in whatever you're studying or measuring. So the role of antibiotics. Penicillin, the first antibiotic, this was discovered in the 1920s. Today there are over 100 types of antibiotics, and you can see how popular they are. And in the United States, in 2015, over 250 million prescriptions were given out. So you can see that they're very popular. However, over 50% of antibiotics was deemed unnecessary. And by 2015, the World Health Organization estimates that there will be about 10 million deaths that can be attributed for drug-resistant bacteria. So this is not a situation that we want to put our society in. We've got to get a handle on antibiotics. Now, how do bacteria acquire resistance? I'm briefly mention how this happens, and it occurs through mutations and through contact with other bacteria that already possess drug-resistant genes. So bacteria acquire resistance in the environment from other bacteria that already contain these genes, and it's through a process involving horizontal and vertical gene transfer. And this is natural and ongoing, so there's nothing we can do. It's constantly occurring. However, as long as antibiotics are overprescribed and overused, the population of drug-resistant bacteria will eventually exceed our ability to treat them with antibiotics, which is dangerous. It's a scary proposition. It's a scary situation for us. So, in my paper, I start off with physical mechanisms and I discuss absorption such as activated carbon, carbon nanotubes, and biochar. And through absorption, carbon-based materials are highly effective because of their properties, such as high surface area, porosity, and strong molecular interactions. There's one technology that was reviewed called carbon nanotubes. This is very promising because it can be synthesized. That means it can be designed to target specific antibiotics. And then PAX, which is short for powder activated carbon, this showed 3 log removal efficiency, and this was for 6 types of antibiotics. And this was done under operating parameters of 2 hours of contact time. But removal of sulfonamide antibiotics was affected by the presence of natural organic matter, NOM. And antibiotic mixtures can be effectively sorbed with functionalized biochar as long as pH is kept at 4.5. So I just mentioned how organic matter interfered with sorption with PAX, but not the same case with functionalized biochar. And then biochar can be commercially treated with coatings such as graphene oxide to enhance sorption performance, such that you get almost 2 times more sorption capacity. Now moving into biological mechanisms of removal, I talk about aerobic treatment, and I go into technologies such as activated sludge, membrane bioreactors, anaerobic digesters, and constructed wetlands. So conventional activated sludge, which is something a lot of us are familiar with, can remove up to 70% of antibiotics, and this was for 5 classes of them. But trimethoprim and lincomycin were observed to only be removed up to 50%, so not as good. And then membrane bioreactors are even more effective. When you compare it to activated sludge, you can get up to 4.8 log removal rates. And then if you base your treatment chain on an NBR, up to 7.1. And then modifying process variables, there were a lot of findings showing that this enhances removal. So when you look at parameters such as solids retention time, hydraulic retention time, these were key drivers. Now going to anaerobic treatment. So for an anaerobic reactor that was followed by a polishing baths and trickling filters, this was shown to be very effective and better than activated sludge for treating sulfa methoxylate and recalcitrant drugs such as trimethoprim. And higher residence time was a significant factor. Now, when a study conducted using anaerobic digester under thermophilic conditions, so this is temperatures higher than mesophilic, they were able to treat up to 5 classes of ARGs, and removal efficiencies were very strong. And they noted that high temperature and the composition of the microbial community played a key role in the degradation. So if you can understand your community microorganisms, this is important for treatment. So you can add things to enhance digesters removal efficiency, such as zero valent irons and natural zeolite. Up to 140% reduction was achieved for tetracyclines, but this doesn't work for other compounds such as copper and CTC. Constructed wetlands. I like this technology because it's not only natural, but it's very inexpensive to build and easy to maintain. It's also common to couple them with microbial fuel cells and biofilm electrode reactors to enhance efficiencies. And one study noted that up to 99.9% for tetracycline compounds was achieved. So pretty good. And a lot of findings in the papers I read were conducted under short-term conditions such as 6 months, 3 months, even a year, sometimes a year. There was one study done for constructed wetlands that involved 10 years looking at 14 classes of antibiotic resistant genes, and they noted that up to 84% removal efficiencies was achieved. So that's pretty good for a long-term study. Now going to chemical mechanisms of removal, and I talk about disinfections such as chlorine, chlorination, UV light, and ozone. And ARG concentrations decrease linearly with higher chlorine dosing, and this was obtained with low chlorine concentrations, and it was shown that almost 1.5 log removal rates could be achieved. And UV light is more effective than chlorination. I mean, UV light is more effective when it's used in combination with chlorination. And it was observed that tetracycline and resistant genes show reductions of 0.6 log removal rates. So for advanced oxidation, I looked into phentan and hydrogen peroxide technologies, as well as using these technologies with UV light. So for instance, for using phentan and UV light treatment, and when you control for parameters such as pH and dosing concentrations, up to 3.8 log removal rates. And similarly for UV light and hydrogen peroxide, up to 3.5 logs of removal. Some conclusions that I'm highlighting here from my paper include microorganisms in wastewater treatment plants are a key driver for antibiotic and ARB removal. So further analysis should be done to discover what are the appropriate matches to specific drugs. Can we look further into how to match drugs to microorganisms? And then adsorption was an effective and economical solution because it provides for smooth operation, high efficiency, and the best part is it has zero byproducts. There's nothing released into the environment, so this should be encouraged more widely. And then more attention needs to be paid to understanding the promotion of resistant bacteria during biological processes, being that our treatment plants are so reliant upon them. So looking at longer solids retention time seems to make the problem worse. It seems to exacerbate bacteria growth. And then constructed wetlands was also another technology I like because it's extremely efficient for treating antibiotics. And when you look at the cost and maintenance, it's very promising. And then there's disinfection technologies. And because this was also important to review because it's commonly used already for drinking water and for at wastewater plants. And several logs of inactivation were achieved. And then when you combine chlorination with UV, guess what? You get more effective treatment and you use less energy. Questions for further review or further studies. Can findings from lab scale pilot studies be appropriately transferred to full scale treatment plants? So a lot of work was done under lab conditions, but can they be used under real world conditions for actual treatment plants? And then what are realistic plant parameters that are generally agreed upon to enhance removal efficiency? So I'm talking about hydraulic loading rates, retention time, pH, temperature. Are there generally agreed upon parameters that can be applied for plants across the world in different regions to enhance removal efficiencies? And then can sulfonamide removal be considered a top priority for treatment? Why? Because sulfonamide is one of those antibiotics that it's very prominent. So it's very used. It's commonly used not only for human health but also for animal medicine as well. Thank you. That concludes my paper. Thank you.